relationship between two variables. So a positive correlation, so it has a positive sign in front of the number, it means that these two variables are moving in the same direction together, either up together or down together. Okay? A negative correlation is going in opposite directions. A negative would be we're going up here, we're going down here, or we're going up here and down here. The examples given are hours spent studying versus GPA. Okay? The more hours spent studying, what do you think happens to your GPA? Yeah, they both go up. Okay? Now, negative correlation. Hours of video games, okay? Hours of video games played versus GPA. And this is a true study, it's a real study. Hours of video games played goes up, GPA goes down. That's a negative correlation. Right? Zero correlation is the statistics did not indicate a relationship between the two factors at all. All right? The scatter plot looks weird. Like this right here, obviously, hours spent studying, grade point average. You can see the more you study, the higher your GPA. Here, video games played, okay? The more video games you play, all right, grade point average, lower it gets. If it was a scatter plot that had zero correlation, all these dots would be all over the place and you wouldn't be able to pick out a line or a trajectory, okay? Some examples here. As the temperature goes up, ice cream sales also go up. So temperature up, ice cream sales up, positive correlation. When workers get a raise, morale improves, positive. A student who has many absences has a decrease in grades, negative correlation. As one exercises more, body weight becomes less, negative correlation. The longer amount of time you spend in the bath, the more wrinkly your skin becomes. More time in bath, more wrinkles, positive correlation. The more one eats, the less hunger one will have. Okay? Eating, hunger, negative. The more kittens you have, the more your batting average in baseball will go up. Zero correlation, unless you're super superstitious, just like me. The experimental method. This is running an experiment, so we'll talk about these things, but we need to know the difference between the independent, dependent, and extraneous variables. These two are really important, right? Independent variable, the purposely manipulated factor thought to produce change in an experiment. It's also called the treatment variable. It's what you are treated with, right? It's probably, in our case, in our example right now, a drug of some sort, right? The independent variable, we give the drug to somebody and we watch what happens to the person in our experiment. The dependent variable, the factor that is observed and measured for change in an experiment, thought to be influenced by the independent variable, also called the outcome variable. So this is what we are measuring for change. It's the person who took the drug. We're going to see what happens to that person, right? Maybe their pupils constrict, right? Maybe their mouth becomes dry, all that kind of stuff. All right, that would be a dependent variable, the change that we are measuring. This is the change administered. This is the outcome. So treatment, outcome. Independent, dependent. Extraneous variable, anything you didn't think of. Anything you didn't think of. A factor or variable other than the ones being studied that if not controlled will ruin your experiment. Let's look at an example. So, the experiment will examine 4 people's ability to throw a ball when they haven't eaten for a specific period of time. 6, 12, 18, and 24 hours. The dependent variable here is the person's ability to throw a ball. We want to see if it changes according to how long it's been since they've eaten. The independent variable, how long it's been since they've eaten. That's the thing that we're changing. All right, and then here are some examples of extraneous or confounding variables. Metabolism and weight of the individual. For example, a 90-pound woman not eating for 24 hours compared to a 350-pound man not eating for 6 hours. Yeah, that's going to play a little factor, right? Ball size. People with smaller hands may have a difficult time handling a larger ball. So, yeah. Or hand size, right? Age. A 90-year-old person will perform differently than a 19-year-old person. Time of year matters. Location of the experiment. You know, is a ball going to travel farther in Denver, Colorado or in Wilmington, North Carolina? Denver, Colorado. And then the person providing the instructions, which is interesting. You know, maybe the person providing the instructions to the experiment one day was moping around, right? Today you will do this and blah, 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 and blah, blah, blah. Compared to the person's like, today you're going to be involved in a ball throwing experiment. You might want to perform better for the person who is enthusiastically giving you the instructions compared to the mundane instruction giver. So, all right, here we go. Experimental method, you got the experimental group. Those are the people who get the independent variable, right? The placebo is a fake substance, treatment, or procedure that has no known direct effects. This can be given to the control group or the placebo control group. Placebo effect, any change attributed to a person's beliefs and expectations rather than an actual drug. It's called the expectancy effect. I mean, this happens all the time, you know. My headache will be feeling better after I take Advil, and I know that Advil doesn't necessarily kick in till 30 minutes after I take it. Or maybe I start feeling alert and awake just by the smell of coffee, because I know that once I drink the coffee, 20 minutes later it gets into my bloodstream, and now the caffeine's in me and I'm pumped up. But I start feeling pumped up when I smell it. I mean, that's placebo stuff. Random assignment, this is just to thwart a little bias. So you sign up for the experiment, you're going to be a subject. You might be randomly assigned to either the experimental group or the control group, or the placebo control group. Double-blind technique, the double-blind study. The experimenter doesn't know who gets the treatment, who gets the placebo, or who gets the actual drug, and neither does the subject. So nobody knows. So there's no bias, right? They're not looking at a certain group and saying, ooh, these people are going to change. Let's look at them. Let's look at them. They don't know. The experimenters don't know who took the real drug, so they have to objectively look at everybody. And then at the end, you find out. Single blind, the experimenters do know, and the subject doesn't. Demand characteristics. So in a research study, subtle cues or signals expressed by the researcher that communicate the kind of response or behavior that is expected from the participant. Again, this is giving demands. Practice effect. This is something you have to take account for in an experiment. What if the person does the same task over and over and over again? Will they get better? Probably. So you have to take that into account. The main effect is any change that can be directly attributed to the independent variable or treatment variable after controlling for other possible influences. This is probably what you're studying. The control group or control condition, those are the people who don't get the independent variable and who we look at to compare the people who get the independent variable to, right? The experimental group is always compared to the control group. Natural experiment, a study investigating the effects of naturally occurring events on the research participants. This is sort of like naturalistic observation. All right, brain scans. This is the positron emission tomography, the PET scan. It is the only one where something is actually introduced into your body. It's invasive. They inject you with a radioactive tagged substance, and it hooks onto glucose, which follows around blood and oxygen, right? And they can watch it travel in your brain. So whenever a certain part of your brain works, it needs more oxygen and blood. And guess what? We can track that as long as we are using that radioactive tagged substance. And that's what it looks like. This is what it would look like on the other side of it. You know, this is about learning language and producing language. So when you learn language, right? Yeah, let me see. This example shows the comparison between subjects learning a new language and performing language tasks after it was well learned. Learning, you're deciding, okay, all right, and listening, and then practiced, you can see that your brain is actually active in a different area. All right. Magnetic resonance imaging, the MRI. This is just taking pictures of slices of your brain. Okay, so actual, it's almost like an X-ray for your brain, but just taking pictures of it in slices. So this is a non-invasive, nothing is put into your body here. Technique that produces highly detailed images of the body structures and tissues using electromagnetic signals generated by the body in response to magnetic fields. I think what they're highlighting here is a portion of the hippocampus. And they can just see if the hippocampi is larger or not. And the research done here was memory according to London taxi drivers. So London taxi drivers, and if their hippocampus actually changes in size because of their extensive knowledge and memory of streets in London. And it did. The hippocampus actually, it was bigger. It was larger than people not taxi drivers. Brain scans continue. This is the FMRI. This is a non-invasive imaging technique that uses magnetic fields to track blood flow and oxygen levels. Okay, so we have examples here, sleep rested and sleep deprived. If we use an fMRI and we watch you know, the brain imaging come in, if you're sleep rested, your amygdala doesn't, you know, it's not as active as it would be or as reactionary as it would be if you were sleep deprived. People who are sleep deprived react emotionally fast, okay? They could, you know, proverbially, bite your head off if you accidentally say something that they don't like. They could just, you know, kind of get in that attack mode, right? Sleep-rested people might not, because their amygdala isn't necessarily inflamed. It's not, it doesn't need as much blood flow to work. Ethics in psychological research. Let's look at this. The American Psychological Association, APA, they come up with the ethical principles of psychologists and code of conduct. You have to follow this when you do research. And then also, they come up with the guidelines for ethical conduct in the care and use of animals, because psychologists do so much research on animals that we have to be ethically, you know, inclined to take care of them. Comparative psychology is the branch of psychology that studies behavior, the behavior of different animal species, and it's huge in psychology. All right, you'll see countless studies with rats that will go over monkeys, dogs, you know, pigeons, tons of animals used in psychology for research. And then there's principles, regulation research with human participants. Human participants in psychological research have to have these things happen to them. They have to be informed, so they have to have informed consent and voluntary participation. You have to sign up for it, okay? Students as research participants. You can use students as research participants. That's what happens usually at a university setting. I remember being a freshman at UNCW, taking a psychology class, going to class every Tuesday, Thursday, and another component of the class was you had to sign up for 9 hours of psychological research. Random experiments done by the psychology department at UNCW. It was required pretty much for me to get an A at least. So 9 total hours of volunteering as a student. The use of deception. You cannot deceive. unless there are no other means, no other ways to justify the means. You can deceive if there's no other way, but don't use deception. Confidentiality of information in their writing, lectures, or other public forums. Psychologists may not disclose personally identifiable information about research participants. You know, it's almost like HIPAA violations there. Information about the study and debriefing. All participants must be provided with the opportunity to obtain information about the nature, results, and conclusions of the research. Psychologists are also obligated to debrief the participants and to correct, you know, anything that could have, any misfortune that could have happened to the participant. We did it. We all done it.